Our session that's going to focus on immunology and cancer. We have three different flavors of engineers represented. Paula Hammond, who's a polymer engineer, thinking about drug delivery and cancer vaccines. Dane Wittrup, who's a protein engineer, working to develop new biopharmaceuticals. And Darrel Irvine, who will be our represented speaker today, tissue engineer who developed synthetic systems to understand the immune response in immunotherapy. So this will be an interesting section on how you can get the body to respond to tumor cells as invaders.
Okay, so my name is Darrel Irvine. I'm going to tell you a little bit about representing Paula and Dane and myself and what we are hoping to bring to the Koch Institute and some of the ways that we're interested in interacting with the other faculty here. And particularly from the standpoint of immunity and developing immunotherapies to treat cancer, which is something I'm very interested in, and so are Paula and Dane. And the premise that this work is based on is the recent evolution of thinking and the resurgence of the idea that there is immunosurveillance of tumors, and that there's now a model in favor of 3 stages where initially tumors are kept in check, or cancerous cells are kept in check by active attack by the immune system, by T cells, macrophages, NK cells, and so on. That this then slowly over time progresses to a state of equilibrium where the immune system is keeping up with growing cells but not actively eliminating tumors that might appear. And then finally, a failure to completely control the outgrowth of transformed cells can lead to escape, where the tumor eventually employs a variety of mechanisms to get around the immune response that's acting against it. So a lot of the things that we're interested in coming from the engineering side are figuring out ways to revert this process and help the immune system overcome this process of eventual escape by tumors. And to illustrate sort of one of the challenges we're interested in tackling, this is a sort of view of the paradigm of vaccination, and one of the things that we're interested in are cancer vaccines. So a typical vaccine that you'd use for an infectious disease would start by injecting an antigen in the periphery with an adjuvant that's meant to stimulate immune cells, in particular dendritic cells, that carry that antigen physically through the draining lymph to the lymph nodes, where dendritic cells activate T cells and B cells and other members of the immune system. And then those activated lymphocytes travel from the lymph nodes, where they start, to the peripheral tissues where they could directly attack a tumor. And a problem with this paradigm, as we now understand it, is that although it works very well for infectious disease, for tumors, one of the major problems is that the actual therapy, the part that you control, is only this initial injection. And so you only have indirect control over what happens in the eventual effector phase via what these dendritic cells do in the lymph nodes. And we now know that there are a variety of ways in which tumors act to suppress the T cells and NK cells and NKT cells that come to the tumor in order to avoid an immune response. So there's a need to be able to act both at the initial phase and then later on at the tumor site to revert these suppressive mechanisms. And this is where we come in. We're interested in both thinking about vaccines that more optimally stimulate immunity or program these cells to how they need to function, and then intervening in the effector phase, helping out the immune system so that it doesn't get beaten by these suppressive mechanisms. And just to highlight a few of them, this is a really exciting time from my perspective to bring engineering thought into these problems, because now we have identified some of the mechanisms by which tumors are acting on the immune system. And there are a variety of them, and this is from a review just summarizing a few of them. You've got the action of regulatory T cells that interfere with T cell effector responses. You have cytokines produced by tumors that are turning off T cells and antigen-presenting cells. And you have so-called myeloid-derived suppressor cells that are secreting factors that suppress immunity. So knowing these players now, and this has sort of been evolving information in the last 5 or 10 years, we have targets that we can go after to try and change what's happening with immunity. And while there are lots of people interested in trying to tackle these suppressive mechanisms, I think what we're really hoping to do here at Koch is bring together both improved vaccination with these anti-suppressive technologies. And where we come in, we're all from the engineering side, and I should say it's unfortunate that we couldn't have some of our immunologist colleagues in this session as well, because what we're really most excited about is the potential synergy between the pure tumor immunologists at MIT and what we can do. Paula brings in expertise in polymer therapeutics. Dane's expertise in protein engineering, and what my lab's interested in, for lack of a better word, we will call immunobioengineering. So to give you some examples, one question is, how do we get vaccines or therapeutics into cells? And this is a problem that Paula's been working on for some time. She has a great strength in synthetic polymer chemistry and characterization in her group, and they've been building new kinds of synthetic materials like these linear dendritic hybrid polymers, where they're really thinking about these as modular building blocks, as illustrated at the top here. You've got a piece that's designed to specifically bind to drug cargoes like DNA, a piece that is involved in breaking these materials out of intracellular compartments and getting them into the cell. You've got, a part that's imparting solubility, and then a part that can target this material to a specific cell. So a ligand that will bind to a target receptor on a tumor cell. And how this all works together then is through a process of self-assembly. So for the case of gene delivery, Paula's group has been looking at complexing electrostatically using charge-charge interactions. You've got a negatively charged piece of DNA that you self-assemble in solution with these linear dendritic polymers that bind on one end and create nanoparticle structures like this just by self-assembly that now have their targeting ligand on the outside, and they're ready to go and interact with a cell. And these materials have a lot of advantages because you can engineer each piece to carry out its specific job. And so she's been able to show targeting to cells in vitro, where you take a tumor cell line in vitro that expresses a certain receptor when there's no gene uptake, and then gene expression is what we're essentially measuring here. They can deliver a gene specifically with the targeting ligand under conditions where there's no binding to the cells in the absence of that ligand. So you can get tumor-specific targeting in vitro. And in vivo now they're starting to look at using this, using whole animal imaging and luminescence and fluorescence techniques to say, can we do this in an animal at a tumor site, for example, and get specific payloads delivered to target components of either the immune system or the tumor itself by having these nanoparticles bind to specific receptors expressed at the site. Where Dane's work comes in is sort of asking the next question. So if you've got this great delivery vehicle that gets things into cells very efficiently with low toxicity, and you want to get them to the tumor, you might ask, what's the optimal ligand so that these things only bind at a tumor site? And this is where some of Dane's work comes in. And his lab has pioneered the use of yeast surface display, which is a technique where you use a live yeast cell as a platform for generating very diverse libraries of proteins, where you have millions or trillions of copies of different mutants of a protein, and then you select out from that library the ones that have the binding properties you want. So you start from some molecule that binds to some target with a certain level of affinity, make mutations in that, and find the version of that molecule that binds with really good affinity, if that's your goal. And then the beauty of the yeast display system is you can characterize things with tools like flow cytometry, and then you can pull the DNA out and make this protein that the yeast was displaying, because the yeast itself is a bag containing the genetic information to make that protein. So it's a really wonderful semi-high throughput technique to obtain new proteins that have unique binding properties. So one of the examples of the dramatic things you can do with this technology is Dane's made single-chain Fv antibodies, so very small antibodies that bind to a target where you go from an affinity. This is basically a binding curve where you're measuring how much of the antibody is still bound to its target as a function of time. And you see that the starting protein decayed in a matter of minutes or hours, and that's typical of many antibodies. And they've engineered this to now have a half-life where after a week, it's still more than 50% of it bound to its target. And the kind of things that you can think about doing then are delivering things to a tumor and having them stay there for days or weeks at a time. And in fact, modeling that they've done has shown that penetration and sequestration of antibodies within tumors in part relies on getting a sufficient affinity so that they can get into the tumor and stay there even in the face of convective flows that might be present. So putting what Dane's working on together with what Paula's technology, you might have new ways to really get things to tumors and keep them there on specific targets. Some work that my lab's been involved in is, again, addressing how do we bring some support to the immune system at the tumor site. And so we've been working on injectable gels that could carry multiple components to bring to bear on the tumor. So, for example, there are current clinical trials going on using what are called dendritic cell vaccines, where you take dendritic cells from the blood of a patient, load them with tumor antigens ex vivo, and then reinfuse them into the patient in hopes of generating anti-tumor immunity. And we've been looking at the idea of delivering those cells in a supporting matrix, a synthetic matrix, that will help keep them at the tumor site, support their activity, and then we could combine them, for example, with controlled release microparticles that are designed to change that tumor environment and fight those suppressive signals that I was talking about earlier, with the eventual goal of then, when the immune response does come in, that instead of being shut down promptly by the activities of all these cytokines and tumor mechanisms, that we could block some of those activities. And there are a number of now molecular pathways that are becoming open that might be interesting targets that, say, interfere with regulatory T cells or interfere with the generation of these myeloid-derived suppressors. And so these are things that we're looking at. So to give you an example of what happens in vivo in a mouse model, we've shown that if you take, we're really trying to leverage the great activity that the immune system has on its own and just support it in a way. So if we inject one of these gels in mice and then look 7 days later to see what cells have come into that matrix by flow cytometry, we're basically looking at the expression of T cell receptor and the expression of a dendritic cell marker. So dots in this quadrant represent T cells that have come into this matrix. And after 7 days, control gels have very few T cells. But if we deliver activated dendritic cells in these matrices, you see this massive appearance of T cells into that matrix. So it's a way to signpost the immune system. In some tumor models, T cells, in fact, fail to get to the tumor. And this is a way we can help bring those cells in by giving them dendritic cells that are located in the gel that might be injected right at the tumor site to bring them in. Another way we can do this, more synthetically tuning what's going on, is to design materials that release factors that might help recruit these cells. And so this is an example where we've been working on microspheres that release chemoattractants. And this is one of the functions that dendritic cells have. But we might try and support them by making synthetic particles that release a chemoattractant, create a gradient in the nearby environment to bring T cells to the tumor site, for example. So this is an example of an experiment in vitro where we take a collagen gel in vitro as a model of ECM with dendritic cells all around, and we're going to release a dendritic cell attracting protein, chemokine, from these particles. And you'll see over time we're imaging at sped up real time, the cells will begin to swarm in after a few hours as the gradient develops. And so we have a way to actively recruit these cells right to a given point. And you can imagine injecting these in the tumor to be bringing immune effector cells to a specific site in the tumor and promote the immune response that way. One last example again from Dane's work. Not only can you think about engineering targeting molecules to bring cargoes to the tumor or to specific immune cells, you can start thinking about changing cytokines that have immune activities. So an example is IL-2 that's been in the clinic for treatment of cancer for some time. But IL-2 is well known to have side effects such as toxicity in the systemic circulation. And it has the problem that although in vitro it activates T cell proliferation, which is what we'd like to get, it also induces activation-induced cell death and promotes T cell tolerance. So there's this two-edged sword to IL-2. And what Dane's group realized is that IL-15, a cytokine from the same family, has many of the same properties of IL-2, but not the downsides. It doesn't promote AICD. It doesn't promote tolerance. And its only limiting factor is that the receptor expression for IL-15 is much broader. So instead of just affecting T cells, you start affecting many cell types in the body. And so they said, well, why don't we engineer IL-2 so that it binds to its receptor and signals in a way more like IL-15. And one of the properties of IL-15 signaling is it binds with very high affinity, and its binding decays over a very long period of time because it's got such a tight binder with picomolar affinity. And so they took IL-2, which has a much weaker affinity and decays from the receptor much more quickly, and engineered it into a mutant that binds and, again, has a decay profile starting from 100% bound to only 40 or 50% bound after a couple of days, much like IL-15. And this then, in turn, has the same signaling properties. So here they're essentially measuring the viability of T cells or IL-2 receptor expressing cells exposed to IL-2 versus IL-15. And you essentially transform IL-2 into an IL-15-like molecule by this transformation. So this is another angle that now you've got an immunocytokine that you're treating some of its downsides by hybridizing its properties with that of another molecule. So I think that's all I want to say to summarize a couple of the ideas here. And really the excitement among us is the opportunity to work with the biologists in the Cancer Center and begin to think in this fashion illustrated by this circle of first doing the immunology to say what are the mechanisms that are dominant and suppressing the immune system in this type of cancer. Then how do we deliver factors that get around that? And if that's not going to do it, what's the next step? Where are we at after that treatment? And I think by working together then we bring some new ideas to the table and some new opportunities. So I think with that I'll stop and we'd be glad to take any questions.
So what is your thought in terms of just how viable an approach? We've heard that vaccines would be a great strategy in defeating HIV, and we've seen where that's gotten us in 25 years. How different a model do you think this is?
Yeah. Well, so my personal take on it would be that now HIV has a different set of very daunting challenges associated with it. But I think it's a question of the timing and the speed at which you can mount the immune response. Because if problems with like drug resistance evolve when we don't kill the tumor quickly enough. And so I think part of it is going to be how can we engineer it such that the immune system destroys tumors very quickly, and couple that with slowing down the ability of the tumor to subvert the immune response by acting on some of these suppressive mechanisms. Now, whether that will be a solution, I think it's a hypothesis, and we're trying to position ourselves to test that hypothesis. What are the limits of what can be done? Some of the existing clinical trials, like with adoptive T cell therapy, where you infuse enormous numbers of T cells into a patient to attack a tumor that show promising responses, suggests that this battle can be won if we get everything right. But
how optimistic are the immunologists that you work with? I mean, I think I read a paper a few years ago about how there had been hundreds of trials, mostly academic, obviously, of cancer vaccines, and all of them, or most of them, had failed for some of the reasons that you're trying to address with your technology. But there's like multiple barriers that you're talking about. Is there a new sense of optimism that's emerging? I mean, there was recently a cancer vaccine approved, but you know,
you're absolutely right. It's a challenging
medical community
problem,
is not that optimistic.
Steve
about it.
Rosenberg was a very high-profile guy who wrote a very damning editorial about the whole field. But I think maybe it has something to do with the way engineers view problems. There's nothing necessarily wrong with the concept. It just hasn't been executed right. And so the question was, how might this go wrong? Well, Darrel laid out some of the aspects of it, that you've got not only the response, but in order to be protective, you have to change the tumor microenvironment.
Right.
And so when problems get messy and there's quantitative aspects to it, that's when engineers kind of like to get involved. So I think that the promise, I have a collaborator who's an immunologist, and he starts his talks where he'll say, picture organ rejection, transplant rejection. That's the kind of fury you can mount against tissue through the immune response. That if you were to sum together all the cells of the immune system, it's, you know, it's the biggest organ, or close to. And so if when we figure out when, not if, when we figure out how to direct that properly, it's going to be very effective. So.
Can I just weigh in a little bit there, Dane and Darrel? I think I agree with both of you, but there was one thing that you didn't say that's important. The complexity, which Darrel reviewed very nicely, of that set of suppressive mechanisms has been underappreciated by the folks who have been pushing forward with cancer vaccines and other forms of immunotherapy. They have either not known about it or ignored it. And in retrospect, it's not surprising, perhaps, that these approaches have not worked so well. And so, in fact, there's a lot of hard science that needs to be done before we'll be ready clinically. And I think the clinicians have sort of rushed ahead before they were prepared. And the consequence is a series of failed trials.
And as an analogy, say, to monoclonal antibodies. So mouse monoclonal antibodies went flying into the clinic, and then people found out that humans raised immune responses against mouse proteins. And so there's an ebb and flow to something that has so much promise. Magic bullets, mouse monoclonals, sounded great, a lot of money got spent, straight into the clinic, and then there was an ebb. And so it doesn't mean that antibodies were a bad idea, which I'm sure you could have found many people to say was the case in the mid-'80s. Just needed to be done right.
I have a question about whether you see this, this kind of vaccine and therapy as being very targeted short-term to a specific condition, or whether, say you successfully fight a cancer using these means, does the patient need to continue to have his or her immune system bolstered in this way? So does it become kind of a chronic therapeutic model? And kind of related to that, the immune system, strikes me as being kind of not a monolithic thing. Everybody has different levels of immune strength, and so is there kind of a broader immunotherapy use to your research?
Yeah, so I think what makes the three of us all want to work on this is the idea that a successful immune response at a primary tumor becomes an immune response that can travel to disseminated tumors and clear tumors throughout the patient, because that's what the immune system does. It can also lead to memory so that you can prevent that tumor from recurring. And that's really a unique aspect of the immune response. So even though some of the challenges facing immunotherapy are very great, the potential reward is a complete and lasting cure. So I think that's what motivates us to work on this. And I think it does broadly apply, but in all the ways that immune responses are important in protecting you from pathogens or autoimmunity or and so on.
And the variability is a place for that hard science that Tyler was talking about to come in. So you take IL-2, for example. It's an approved drug. Only about 15% of the patients that take it get any benefit whatsoever. 85% get nothing. Why? What's the difference? If they knew, they wouldn't give it to those 85% because the side effects are terrible. So there's a need for understanding the sources of that variability.
And in terms of how broad these kinds of applications can be extended to other immunotherapy problems. Taking, for example, the material science end, which is where I work, there are a number of inherent problems in being able to deliver either drugs or DNA to these kinds of cells. Dendritic cells are particularly challenging. So by looking at and focusing on these problems, we can actually learn a great deal, which will help us with other immunotherapy applications. Learning how to design, for example, a carrier, which can be very selective in where it's directed. Learning how to allow DNA or whatever it is we're trying to deliver to get out and to be effective in the cell. And those kinds of problems that we're studying as engineers can then be applied to a broad range of problems, including a number of problems in cancer.